Great. Well, today is April 1st, so I don't have any April Fool's jokes for you, but do have some science. So we're going to talk about mapping EQTLs and the HLA genes at single cell resolution. So here we go. So as an outline, first I'll kind of motivate why we're interested in studying gene regulation in the HLA and why we want to do it at single cell resolution. So as you may recall, the HLA genes are these genes that are very important for the immune response. And that's because their main job is to present antigens or like short peptides to T cells. And then the T cells can recognize those antigens with their T cell receptors and become activated to trigger an adaptive immune response. Very important to kind of differentiate between self and non-self. And so to dig a little deeper, the HLA class 1 genes present typically to CD8 T cells, and then the HLA class 2 genes present to CD4 T cells. And an important thing about these genes is that the HLA gene family is very polymorphic. So there's many different versions of these alleles across individuals. And so scientists have done genome-wide association studies looking for associations between genetic variants and traits, and turns out that the HLA locus contains the most number of genetic associations with diseases genome-wide. And then in terms of immune-mediated diseases, such as autoimmune diseases and infectious diseases, it turns out the effect sizes within the HLA are typically larger than all other loci combined. So as you can see here, for HLA-B27, it's very highly associated with ankylosing spondylitis and there are other HLA disease associations. And so exactly how these different HLA variations lead to disease is not fully understood, but there are several possibilities. And so one of these is a conventional thought, which is that there are coding variants, so like these red triangles within the HLA genes themselves that code for protein changes in the protein of the like the HLA molecule, and that may affect binding to certain antigens, better presenting self-peptides, affecting the quality of antigen presentation. And a less well-studied hypothesis is that regulatory variation may play a role as well. And so, for example, there could be these regulatory variants like this blue variant here that affects things like transcription factor binding that might affect not the quality of the HLA molecule, but rather like the quantity of HLA expression and antigen presentation. And so there has been some prior evidence that HLA expression levels are important for disease. And so we really wanted to kind of understand and kind of tease apart these HLA regulatory effects. So that brings me to the concept of an EQTL, which you may be familiar with already, but I'll just kind of review it. But an EQTL, or expression quantitative trait locus, is basically a genetic variant that affects gene expression levels. So another name for these regulatory effects. So you can see there's a blue EQTL here that affects the expression levels of this red gene G, where certain individuals who have, say, 2 copies of the C allele will have higher expression of G compared to those with 1 copy or 0 copies, as an example. And so traditionally, EQTL studies have been done using bulk RNA sequencing to try to look at associating a gene's expression level with genetic variants across a bunch of samples or individuals. And now with single cell data, we can now look at this at higher resolution by separating out the cells by their individual cell types. And that can help us find EQTLs whose effects might be masked when all the cells are averaged together. So as an example here, each dot is a cell, and if we take, for example, all the B cells, the green cells, there may be an EQTL that's present when we look at this cell type by itself, but is not present in the other cell types, so we would call that a cell type dependent EQTL. We could also find dynamic EQTLs or cell state dependent EQTLs that vary in their strength across cell states within a given cell type. So in this example, all these teal cells are T cells, but then you could see that in this example, there's an EQTL effect that gets stronger in these like darker T cells compared to the lighter T cells. And so to be able to really understand regulatory effects in the HLA, we really needed to map the EQTLs for the HLA genes and set out to do that in multiple different cell types and tissue contexts. And so there have been some previous efforts to do this, but they've mostly been in bulk, in cell lines. And so we wanted to do this using single cell data from multiple data sets. And so to do this, we used the following set of data sets, these 4 previously published studies that have data from PBMCs as well as synovial tissue from RA patients, rheumatoid arthritis patients, and intestine. And altogether, this data set contained over 1,000 individuals and about 1.36 million cells altogether. And importantly, all of these data sets had both genotyping data as well as single-cell RNA-seq data. And so that brings me to the second part, which is how do we actually measure HLA expression accurately? And the reason we want to be able to measure this accurately is because, again, in our EQTL analysis, we basically want to test this association between HLA expression and genetic variants. And so the dependent variable is HLA expression. And so we need to make sure we estimate that accurately to get good results. And for technical reasons, this is especially challenging for the HLA genes. And I'll get into that, into why that is. And basically, the problem is that you can run into this issue because there's only one version of the HLA alleles in the reference genome, which can cause a bias when you're looking at HLA variants coming from a lot of diverse populations. So if you imagine you have two different individuals, one of them is homozygous for this orange triangle allele, and the other individual has this blue rectangle allele for HLA-DRB1, and say, for example, that the reference genome has the orange triangle version. What can happen is if you're trying to map the sequencing reads from the orange individual, their reads will map well to the reference because they have the same version as the reference, and you'll get accurate expression estimates for their HLA expression. However, for the blue rectangle individual, when you map their reads, you can get unmapped reads or mismapped reads leading to underestimations or incorrect expression estimates. And so to kind of overcome that problem, we had to develop a new pipeline, which we're calling single-cell HLA BERS. Stands for quantifying single-cell HLA expression with a personalized pipeline. And the intuition behind this pipeline is that instead of using the single reference genome, we actually build a personalized reference genome for each individual. And so it's a two-step process. The first step is that we using the genotyping data for each individual, we can impute their HLA alleles using this existing method called SNP to HLA, and grab those HLA sequences from this database and add them to the reference genome to personalize the reference genome for each person. And then we we use Star Solo, but one could use Cell Ranger too, but we use expression read counting algorithm to be able to then output a genes by cells matrix, which is like what Cell Ranger would output, except for now the HLA genes are estimated much more accurately. So we did a bunch of different benchmarking analyses to show that personalization does actually improve HLA expression estimation. So in this result, I'm showing you the standard pipeline is when we use a single reference genome, whereas the personalized pipeline is when we personalize the reference. And so we're looking at individuals who have 2 copies of the reference in red, and then increasingly dissimilar alleles as the reference in the light blue and dark blue. So you can see for individuals homozygous for the reference allele, when we personalize the reference, their HLA expression doesn't change very much, as you would expect, because their allele already matched the reference. However, for individuals where we added new personalized sequences to the reference, you can see that their HLA expression estimates actually change with the improved pipeline. For some individuals, even more than like 50%. For people whose alleles are very different from the reference. And so now with our more accurate HLA expression estimates, this allowed us to map EQTLs for the classical HLA genes across these different cell types. So we focused mainly on myeloid cells, B cells, and T cells, and we did an analysis per cell type, kind of a pseudobulk analysis where we took each major cell type and aggregated the cells per sample to create a pseudobulk profile for each individual. And then we combined all the cohorts into one single model and performed the regression analysis to be able to identify EQTLs. And so our results kind of looked like this for each cell type. Here, it's a Manhattan plot where each dot is a test for each genetic variant on the x-axis for its association with each HLA gene's expression on the y-axis. The y-axis is the significance, and then the colors are the different HLA genes. And so for each HLA gene, I've labeled the lead EQTL variant with the strongest signal for each gene. And so if we take a look at one example, so this RS310 SNP, this was the lead EQTL for HLA-DQA1 in myeloid cells. So we can take a look and see across different individuals here and across the different cohorts, which are the different colors, we see that people with more copies of the G allele have higher DQA1 expression, and it's consistent across cohorts. And interestingly, this particular variant commonly co-occurs with this HLA-DQA1 0301 allele, which is known to be associated with type 1 diabetes and celiac disease. So it could be that this disease association could be tagging both like a coding effect or a regulatory effect, or both. So it kind of ties into that this variant could be kind of mediated by two potentially different mechanisms. And so now we, as an example, another example, here's an example of a cell type dependent EQTL for HLA-D-RB1. So you can see in this example, this variant has a really strong effect in B cells, but then doesn't have a significant effect in myeloid and a much lower effect in myeloid cells and no significant effect in T cells. So it kind of shows how important it is to consider cell type in EQTL analyses. Great. And so now we can talk about how do we integrate multiple data sets to define cell states. And so everything up until this point, we could have basically done with bulk RNA-seq since we used a kind of pseudo-bulk model in the previous results. But now we wanted to kind of actually look at each single cell individually and model incorporate cell state into the model. And so single cell EQTLs, like I mentioned earlier, they can capture these dynamic regulatory effects changing across cell states. And one key question in terms of how to actually formalize these models is how to define the cell state variable. So we need some way to be able to label the cells or order the cells along some sort of variable that can be captured in the model. And so in our study, because we had 4 different data sets, we wanted to be able to define cell state in a common way across these different data sets. So the problem here is that when we're analyzing these multiple data sets together, it can get complicated and there are these unwanted technical effects. As an example, you can see here the 4 data sets. If we look at just the T cells and we run kind of a standard dimensionality reduction pipeline, you can see that the biggest variation is actually which data set the cells came from rather than what T cell type they are. And so what we're really interested in is the cell states. And so the solution to this is that we used this approach called single cell reference mapping, where we can align and integrate cell states from different sources into a kind of harmonized lower dimensional embedding. So we used a method we developed previously called Symphony to be able to define a reference kind of coordinate space using the 2 PBMC data sets, and then we mapped the tissue data sets onto, or it was the other way around. We developed the reference using the tissue data sets and then mapped the PBMC data sets on top. So you can see here, now we have all the T cells from the multiple different data sets all together harmonized in the same space. This allows us to make the single cell data from different data sets comparable. And so here we have those cells colored by their cell states. So you can see similar cell states across data sets align, which is what we wanted, which is great. So that was showing an example with T cells. We did the same thing with myeloid cells and with B cells. So we could take the multiple different data sets, align all the cells within the same cell type, and look at patterns of HLA expression across these different cell states. So you can see, for example, in B cells, these kind of purple areas are the plasma cells and plasmablasts, whereas these which have lower HLA expression compared to these more yellow regions, which are the naive and kind of non-plasma B cells. And so now that we have all the cells in a harmonized lower dimensional space, we could actually use those principal components, those like harmonized principal components as variables within our model to figure out whether cell state influences these EQTL effects. And so, yeah, so that brings me to the last section, which is, does cell state impact the genetic regulation of HLA expression? And the short answer is yes. And it turns out, especially for the HLA-DQ genes, cell state has a really strong effect. And so to kind of get at, methodologically, to kind of get at this, we used a negative binomial mixed effects model where we modeled the UMI count of each HLA gene in each individual cell as a function of the cell's donor-related effects, individual cell level effects, as well as interaction terms between these harmonized principal components, which capture cell state, and the genotype effect. We also added random effects to control for repeated sampling of cells from the same donor or batch. And so this gives us a result that looks like, this model kind of gives us a result that looks like this, where each dot is a different test of an HLA EQTL, and the y-axis is telling us how cell state dependent that EQTL is. So you can see that a lot of HLA DQ genes pop up as the most cell state dependent across these cell types. And I'll show you a couple examples. So here's one example of this RS310 SNP for HLA DQA1 in T cells. And so you can see from our model, we can estimate kind of a total EQTL effect, taking into account the main effect of genotype plus all of the interaction terms between genotype and the different principal components which capture cell state. So for each individual cell, we have like a beta total for how strong that EQTL's effect is within that cell. So as you can see in the blue cells, the EQTL doesn't really have a strong effect, whereas in the orange cells, you can see that there is a much stronger EQTL for those cells. So this was with data coming from the synovial tissue data set, and we found that even in T cells from the blood data set, the same effect replicates in a completely different context. And so if we map this on to like what the actual T cell states are, we saw that this stronger effect was happening in more cytotoxic T cell states compared to naive T cell states. And we thought this was a really interesting example because usually people think of T cells as the cells that recognize HLA, but here we're seeing that T cells themselves express class 2 HLA genes and seem to internally regulate how much HLA is expressed, depending on their cell state. And so that is unique to humans and hasn't been really seen in mice. And so that could be a source of interesting variation across people in terms of their immune response. As like a last example, this is another one in myeloid cells where we can see that this EQTL has like a stronger effect in these monocytes in the orange cells. The genetic effect of the EQTL is stronger compared to the blue cells, which were the dendritic cells, which had a much higher expression, but the EQTL's effect was lower. And so it could be here that dendritic cells are professional antigen presenters, so their expression of HLA DQ is always high, whereas maybe there's been more evolutionary lenience for monocytes to have differences in terms of their HLA expression across individuals. So as a summary, we set out to understand HLA genes, which are critical for the immune-mediated diseases. We developed single-cell HLA PERS, which is a personalized pipeline to quantify HLA expression in single cells. By combining 4 different cohorts, we could map EQTLs for these genes in diverse cell types, and we were able to find dynamic HLA regulation. And regulation of these HLA expression levels may contribute to potentially disease-relevant and biologically important variation across individuals in terms of immune responses. And if you're interested in learning more, here's a link to the manuscript as well as the personalized pipeline single-cell HLA PERS. And with that, I want to thank all of the lab mates and collaborators who helped contribute to this work, as well as funding sources, and would love to take any questions.
Yeah, thank you for a wonderful talk. Let me just check if there are any questions from the audience. Okay, I'll ask one till the time maybe they'll write it down. So my question is, so how does, since like HLA has polymorphism, so how does these different variants of the HLA gene affect different cellular pathways in the immune system? And how does it affect the cellular state?
Yeah, it's a great question. Kind of why would it be helpful to have such polymorphic genes across individuals, maybe is what you're asking.
Yeah.
Yeah, so I think the hypothesis there is that as humans and as a species, we've evolved over time through various selective pressures due to infectious diseases, and having a highly diverse HLA repertoire, presentable antigen repertoire helps us to be able to, as a population, have kind of herd abilities to present novel viruses or a diverse array of viruses and bacteria that might cause infectious diseases. And so as a species, it's beneficial for there to be a high degree of polymorphism in these genes to be able to protect us generally from infectious disease sources. Because mechanistically the HLA are kind of taking antigens, either self-derived or from exogenous like infectious sources, and presenting them to T cells. And so only if we can be able to see novel antigens and develop an immune response to these infectious sources, and the HLA is sort of the main protein that kind of determines whether these peptides are able to be seen by the adaptive immune system.
Okay, great. Yeah. So there's one question from Ohana. She says it's a very interesting study. And she's asking, do you think this pattern of EQTL regulating cell type specific expression might occur to other polymorphic genes in the genome, or is something particular of HLA?
Yeah, that's a really great question. And I think it's definitely not HLA specific. There are a lot of EQTL and single cell EQTL studies kind of going on at a genome-wide scale right now. And we wanted to, but our lab kind of has expertise in the HLA, and so having developed some of these like HLA imputation algorithms in the past, and so we really wanted to be able to kind of focus on the HLA genes and give them kind of the care that they deserved in terms of being able to estimate their expression accurately and kind of map the EQTLs for those genes specifically was kind of a big undertaking in and of itself. So that's kind of why the paper focuses on HLA genes, and it was its own project. But there are definitely a lot of single cell EQTL mapping projects going on for whole transcriptome EQTL studies. I think with the 1K cohort is a great example of a kind of big cohort that folks had kind of done a more broad scale analysis for. And yeah, so for other polymorphic genes, like the KIR genes or other genes, I assume that kind of having a better ways of measuring the expression would be able to help improve accuracy for EQTL detection in those genes as well.
Yeah, there's one question from Kathy. So she's saying, thanks for the great talk. She's curious how we can attempt to combine EQTL data with data on the ability of HLA coding variants to present antigens. It seems like the combination of the two could be important, but also a huge combinatorial space.
Yeah. Is the question kind of how can we combine them to better understand kind of like variant effect mechanisms, things like that? Yeah, I think, one kind of challenge we ran into with this exact question was, when we found these signals, these EQTL variants associated with HLA expression, it was really difficult to like fine map those individual variants to like the specific like promoter or enhancer that might be causing these changes in expression, because the HLA has a lot of linkage disequilibrium. So a lot of variants in the HLA tend to get inherited together. And that causes some challenges with the statistical models where it's really hard to tease apart individual kind of areas of where the variants are physically, in this gene sequence. And so I think it's like, for example, with this HLA-DQ A1 EQTL I showed for T cells, or for myeloid cells, the lead variant, it's really hard to kind of say for sure whether that is tagging a coding effect as well for the disease associated part. But we do see that it has an effect on expression. So I think it's really hard because the HLA is a part of the genome that has such high linkage disequilibrium to really be able to tease apart exactly the difference between the coding and the regulatory effects for that variant as an example.